Okay, so now you have the molecular mechanisms for sodium reabsorption in the proximal tubule. 67% of it is by those mechanisms that are circled in orange. 67% of the water that we reabsorb is following this 67% of the sodium by this paracellular pathway. Okay? You now have the concepts for the rest of the day, but the details are going to be different. Are there questions about how sodium is reabsorbed in the proximal tubule? Let's go to the ascending limb of the loop of Henle. 25% of the sodium and 0% of the water is going to get reabsorbed here. What do we need to know? What is this? Sodium potassium ATPase. Hydrolyzes ATP, pumps sodium out from the lumen to the peritubular space. We have the sodium proton exchanger, same one we saw in the proximal tubule. It's also it's moving sodium into the cell. But here is a key difference between the proximal tubule and the ascending limb of the loop of Henle. This transporter in the apical membrane is expressed only in the apical membrane of the ascending limb. This transporter has a name that is actually very helpful. It's called the sodium potassium 2 chloride cotransporter. That is its actual name. So its actual name, sodium potassium 2 chloride cotransporter, tells you what it does. It cotransports sodium down its concentration gradient and brings potassium and chloride along for the ride. We don't really have to talk about anything else on this slide because we'll talk about this guy later when we're talking about bicarbonate. It's not related to sodium. We're not going to talk about this because it's not critical. You'll notice water is not moving, right? Water is not moving. That's a cross-out sign. Water is not moving into the cell and is not moving paracellularly because this is a tight junction, a non-leaky epithelium.
The sodium potassium 2 chloride transporter is only in the loop of Henle.
Only in the apical membrane of the ascending limb of the loop of Henle. But now you should be able to see how the ascending limb of the loop of Henle pumps sodium. It transports it across the apical, and then it pumps it out basolateral. Okay? It's going to get easier. Distal tubule. We're walking our way through the nephron. We'll come back to the descending loop of Henle later. So proximal tubule, descending loop of Henle later, ascending loop of Henle that we just did, distal tubule now. What is this? That's the sodium potassium ATPase. Which membrane? Basolateral. It's always basolateral. What is this? Name it. And I'll just tell you, its name makes sense. It's the sodium chloride cotransporter that is expressed in the apical membrane of the distal tubule. Is this protein the same as this protein? No, it's a different protein. It's a different transporter. It obviously shares some properties. But this is the sodium potassium 2 chloride cotransporter, and this is the sodium chloride cotransporter. Different proteins, got different names that share some similarities. So, yeah, I forget, 3% or so of the sodium is recovered by this mechanism. Sodium moving across the apical membrane and then pumped out basolaterally. And now finally, we'll talk about the collecting tubule. And this image is of the cortical collecting tubule, but the medullary collecting tubule has the same mechanism. So in fact, we should just talk about the collecting tubule. Let me use a different color. The collecting tubule. What is this? Sodium potassium ATPase is expressed there too. So it's expressed on the basolateral membrane throughout the nephron. What is this? A sodium channel. It is not a voltage-gated sodium channel. It's got a name called ENaC. This is ENaC. What does E stand for? Epithelial. Epithelial sodium channel. So sodium flows in and is pumped out in the collecting tubule. Now there's this interesting thing here that you will use in your problem set. In the collecting tubule, there is this other thing called the potassium channel. And so this is acting like an exchanger. Every time sodium moves in, potassium moves out. But it's not the same, it's not an exchanger, it's not an obligatory exchanger because it's two different proteins. But it acts like an exchanger. So when sodium flows in here, potassium flows out there. And you'll use this in the problem set. But, so we're not talking about potassium too much. Sodium flows in and pumped out. And we have just recovered 99.5% of the filtered sodium by these mechanisms. And I do want you to know the things that are circled. So it's the details are what's the word? Imposing, but the concepts are straightforward. And so if you learn the concept of apical and basolateral transport, and then you fill that in with the details, it will fall into place. And the problem set is going to help you fall it into place. Okay? But I do understand that this is intimidating amount of information. I like using this image because it's graphical and it helps me see what's going on. If you would prefer all the information is here as well in text form. I'm not going to go through it. This is the text version of the graphic that I showed you in the previous slide. So for example, on the proximal tubule, we have to think about the sodium glucose cotransporter. We have to think about the sodium proton exchanger. We have to think about the sodium potassium ATPase on the basolateral membrane because it's present everywhere. So this slide can help you. If you take this slide and map it onto the slide with the images, you can come at it from both ways. text information, graphical information. I think it's, using the combination will be helpful for you understanding what's going on. So let's not go through the stories again text-wise, because we just went through it in gory detail graphic-wise. Questions? Let's skip this one. It basically says stuff that you already know. Sodium is actively pumped across the epithelium, it's pumped across the basolateral membrane, and it flows across the apical membrane. And then it diffuses. I guess this is the point. Remember, we have gotten sodium from here to here by all these transport mechanisms that you should start to be able to recite. And then the sodium is simply picked up by the blood to get recycled from the kidney back to the rest of the body. Okay, so sodium transport, sodium pumping, diffusion, 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 or flow. Let's do a couple of clickers. Which of these transporters is helping reabsorb sodium in the proximal tube? Good. They all are. Which of the following describes how water gets across the epithelium in the proximal tubule? Mostly correct answers. B is the only one that's correct in this case. Water flows passively because of osmosis. It's flowing towards the solute. And the solute is being pumped. In this case, the solute being sodium. Okay, I think we have some more. Which of the following are true? Okay, we're talking about sodium, so I'm trying to apply your knowledge of GFR and what you just learned. Clearance of sodium is lower than GFR because it is filtered and then reabsorbed. Sodium is not secreted, it is reabsorbed. Sodium is freely filtered at the glomerulus and is reabsorbed. That's what we've just been talking about. It is not secreted. Secreted means it's being pushed back into urine, and that's not what's happening. We are reabsorbing. Let's get this language straight. Secretion, in this case, leads to excretion, and reabsorption leads to recovery and less excretion. Okay, we can do glucose because it's simple. It's ways, I needed to do sodium first for many reasons, but glucose is simpler. And you already know some of it. Where is glucose reabsorbed? What segment of the nephron accounts for glucose reabsorption? The proximal tubule. We don't have to think about any of the other segments of the tubule. We own this, so that's easy, number one. Glucose is absorbed by the proximal tubule. This slide makes a deal out of the early and the late proximal tubule. You don't have to make a deal of that. So let's just look at the mechanism. Now remember, we're thinking about glucose. This is the lumen, this is the epithelium, this is the peritubular space. We're trying to get glucose from here, because it was filtered, back into our blood where we can take it back to our brain where we need it. We don't want to excrete it. We work pretty hard to get that glucose. We want to conserve it. What is this? Sodium, potassium ATPase. We can't ignore sodium, potassium ATPase even here because of the sodium glucose co-transporter here. Sodium, going down its concentration gradient, is pumping glucose into the cell. This is secondary active transport of glucose, driven by the sodium gradient, which was created by the sodium pump. So we've now gotten sodium from the lumen into the cell. How do we get sodium? Sorry, we have gotten sodium, but we have also gotten glucose from the lumen into the cell. We get it from the cell into the peritubular space by a glucose uniporter called GLUT1 or 2. So glucose is secondary active transport pumped, apical, and then passively flows basolateral. And is picked up then by the capillary, just like sodium was. This book makes a big deal about, so what does SGLT stand for? Sodium glucose transporter. So the names are actually pretty helpful. Sodium glucose transporter, and there's two forms. There's 2 and 1. Don't spend your cognitive load on that differential. It turns out that there are 2 types of sodium glucose cotransporters, and they each have slightly different properties. What I want you to know is that there are sodium glucose cotransporters apical, there are sodium pumps basolateral, and there are glucose uniporters basolateral. That's the main thing. I don't want you to spend energy memorizing where GLUT1 is and where GLUT2 is, or where SGLT1 is and SGLT2 is. Sodium glucose cotransport, glucose uniport. Pumping of glucose, passive transport of glucose. All of your recovered glucose is by this mechanism. If the glucose gets past the proximal tubule, so here's the text version of it. Here's the text version of what the graphic previous showed you. If the glucose gets past the proximal tubule, you will excrete it and lose it. And that's what happens in diabetes. So here's a slide. Here's some data. This is actual data. I mean, it looks like a cartoon, but it's actual data. So here is a graph of either filtered load, reabsorption, or excretion, depending on the color. Okay, so it's in milligrams per minute. this is the normal range. This is the normal range right here of glucose in plasma. So right now, you are semi-fasting. You probably had breakfast, but so you're not really fasting. But fasting glucose is somewhere between 80 and 160 milligrams per mil. So this is, or milligrams per deciliter. You are excreting 0% of your glucose because you are reabsorbing all of it. But if the glucose concentration gets above about 250 or 300 milligrams per deciliter, which happens in diabetes, then the mechanisms of the proximal tubule, the sodium glucose cotransporter, the glucose uniporter, working as hard as they can, can't recover it all. And you start to, this is, the red line is the recovery of glucose as a function of the glucose concentration. So the higher the glucose concentration, the higher the rate of reabsorption, up to a maximum. Because at a maximum, your transporters are working as hard as they can. They're saturated, they're doing everything they can do. And if we go above that, we start to see, so this is reaching its maximum of reabsorption. And if we reach a maximum of reabsorption, we start to excrete it. The kidney has no mechanisms for adding on additional glucose recovery after the proximal tubule. It just doesn't do it. So if the glucose gets past the proximal tubule, it's going to end up in urine. And that's what happens in diabetes.
Can this happen, if the person, for example, on that day ate I don't know, 600 grams of sugar, and a normal, they just had a day where they just, for something, once it did.
I suppose that if you went a little crazy and just ate nothing but, but remember, it's not just, we make sugar out of carbohydrate, and we make sugar out of protein, but in principle, I suppose it's true. If we were to overload our bloodstream with glucose, we would excrete some of it for a short period of time. What happens in diabetes is that it's chronic excretion of glucose. Okay, so we'll skip it. Maybe we'll come back and do a clicker after on Thursday. On Thursday, what we're going to do is, I just want to give you a heads up. On Thursday, what we're going to do is we're going to look at kidney cells. We'll do, what's the word? Histology, thank you very much. We're going to do kidney histology in lab on Thursday. You'll see the different segments of the kidney in pictures like this on Thursday. Okay, so we're done for today. Think sodium, think glucose, think mechanism, and we'll pick up with bicarbonate and potassium on Thursday.
Okay.